Guess what? You're getting ready to do another electrophoresis gel. Can you believe it? Yeah, I know. You're probably saying right now or gritting your teeth, oh no, another one. But guess what? There is another reason to do a third electrophoresis here. And what you've done up to this point is parental DNA. So you have ran a sample, and this sample has been a child's sample most of the time, and then you have tried to compare the mother and the father DNA bands to the child because you now understand that we receive our DNA from both. Well, in this example is more of the forensic side of the house as far as the crime scene goes. So what's going to happen in this scenario? It's going to be another simulation that doesn't really use any actual DNA, but different samples at least that have already been prepared for you. What's going to happen is that one of the lanes that you're going to pour this time will be a crime scene. So this is maybe a body fluid or blood that they have obtained from the crime scene. They've brought it into the forensics laboratory, and they want us to analyze the DNA fingerprint for the crime scene sample that they have submitted. And then in addition to that, you're going to see some suspects here. And I think that this lab is going to give you 3 different suspects that you maybe have, or not you, but the police have in custody. So we need to figure out which one of the suspects will actually match the crime scene. So for that reason, I think that this lab experiment is going to be a little bit easier for you. So you're going to follow basically the same steps that you have followed before in the past with our electrophoresis gel. It's still going to use agarose. We have a certain percentage that we need to make. We're going to let it cool down to 60 degrees, and then we're going to pour it into the molding tray. When we pour it into the molding tray, then we can actually inject our samples just like before. There's going to be no difference. And one of the lanes will be another DNA ladder. We always like to run DNA ladders when we run samples because this gives us an idea of the size of the base pairs that we're working with, whether it's our crime scene sample or our suspect sample or whatever we're running. So in the very first lane, you'll typically see the DNA ladder, and this lab experiment will tell you which band represents what size base fragment, period. All right, so that one's going to be an automatic. We're going to run it. You're going to see it. There probably will be 6 or 7 bands that will be there. So there you go. The other 4 samples, again, crime scene and suspect 1, suspect 2, and suspect 3, will be ran in separate wells. So again, we can run multiple DNA samples in one gel if we have to. But here's the difference, and I actually think that this is going to be a little bit easier for you. Because if this is the crime scene, then we know that a certain particular type of banding will happen. And then let's say that this is suspect number 1. Well, if suspect number 1 doesn't really show the same exact banding pattern, then we know that suspect number 1 is not the person whose DNA sample they found on the crime scene, right? So if we run suspect number 2, and let's say that it, again, is different, then we know suspect number 2 is not going to be the person that they have obtained sample-wise on the crime scene. And there is a possibility that they might give you sample number 3, and it has completely different banding as well. So maybe none of the suspects will match the crime scene. And if so, then that's great for the suspect. They're free to go, right? At least for now. But what we want to compare here is an identical match, and this is where this electrophoresis is a little bit different. All right, there is an identical match because we know now that people have their own DNA fingerprint or fragmentation pattern. So if we find a sample on the crime scene and it fragments in a certain way, then whosever DNA sample that is will also fragment in the identical way. So this is not a comparison between 2 or more different lanes to figure out who's the mother, who's the father. Basically, it's a hybrid of the 2. This is a truly look at the crime scene sample, look at the suspects, see if any of them perfectly match. And if they perfectly match, then guess what? You've just helped maybe get a little bit closer on who was at least present on the crime scene. Maybe not necessarily prove that they did the crime, but they were at least present. And that's kind of half the battle, or part of it at least. So this is the lab that you're going to see in your lab kit or your packet when you download it and begin to review it and read it before the lab, and it's called DNA Fingerprinting by Pcr Amplification. So if I scroll through and go maybe to the first type of background page that they want you to read, they're going to kind of give you some background information. First, we've probably talked about DNA at this point, right? Deoxyribonucleic acid. So DNA, we now know, is present in every single thing that we do. If it's living, then it probably has a DNA sample that I could extract, and I could run them, and I could break them up into fragments and put them on an agarose gel and electrophoresis, just like what we've done previously in our 2 other attempts with electrophoresis at this point. So we know that this is genetic information, and on a crime scene, we can obtain that DNA, and we can obtain that DNA through a number of different kind of outlets. One, if there is a hair follicle or a strand of hair anywhere that we have discovered that we have found, we can extract DNA from this, believe it or not. So just one hair strand, that's really all that we need. We can bring it into a lab, we can amplify it, we can make more of it, and then we can analyze it. A biological stain, that could be semen or blood, and we could extract that blood or semen sample off of the piece of fabric or the carpet or maybe off the plaster of the wall. Who knows where it's located, but we can extract that DNA from that sample. Again, we can bring it into the lab and we can amplify it and we can replicate it, we can make more of it, and then that way we can analyze it a little bit better. And finally, skin cells. If there are skin cells, or basically if I take a piece of tape and put it on the outside layer of your skin and rip it off, then I've got some skin cells, and I can take those skin cells and I can bring those into a laboratory as well. And I can basically analyze it, replicate it, and do everything that I need to do with a DNA analyzer. Suspect number 1 and suspect number 2, these are people that are typically brought in for questioning, right? So you actually have them in person, and the easiest way and probably one of the better ways to extract their DNA, they listed it here. Number 1 is a blood draw, but number 2 is also a cheek cell sample. So you can either have a phlebotomist to draw blood from them, and we can utilize that blood and extract the DNA and begin to analyze it and replicate it, or you can basically give them this long piece of plastic, basically, that has a little bit of tape or sticky stuff on the end, and you just basically have them to rake that across their cheeks, and those cheek cells will basically slosh off, and it will capture those cheek cells, and that's no different than any other cell. So you can take those cheek cell samples, you can preserve them, you can transfer them to the laboratory, and then there we can extract the DNA from those and replicate them and analyze them without a problem. So no matter what case that you're working with, whether it's a blood draw, whether they're cheek cell samples, whether it's from hair or a stain or a skin cell, there's always some common treatment that we do with these samples that are donated to the lab or given to the lab. And number 1 is that we have to treat it to release the DNA. So this DNA is typically incorporated in cells, and again in the lecture module, if you've not made it to that point yet, you'll start to learn about where that DNA is located and how it's organized. But we have to release it. We have to pop those cells open, and we have to extract that DNA so that way we can do stuff with it. So this is going to be the number 1 step. We have to treat it to release the DNA, and that's kind of what they're showing here. So once this DNA sample is liberated, then you've got this into a tube, and now you can do things with it. Well, it probably is a really small sample, especially if it's found at the crime scene. So in the next step, what we have to do is something called PCR. And PCR stands for polymerase chain reaction. And I'll show you where that's at in just a minute in the background information, in case you want to spell it out the proper way. But polymerase chain reaction is going to amplify, that's what we call this, amplify various parts of your DNA sample. And amplify just means make a copy of. Okay, so this is a way that we can take a very small DNA sample and we can copy it and copy it and copy it and copy it over and over and over again until we get a large enough sample in the lab that then I can do things with and put it on an electrophoresis or put it on an agarose gel and do other things, whatever I need to do with it. This number 2 step has a lot of reagents that come along with it. And these reagents are necessary because, number 1, you have released the DNA, but you haven't separated it yet. So you need to separate the DNA to expose the base pairs. You then need something to go onto the DNA strand as almost like a starting area for the replication to take place. You then need to give the DNA enough base pairs so that it can take those base pairs and begin to replicate itself and make that second chain of DNA that we've talked about in lecture so far. So these are the areas that we need to really pay attention to because all of these reagents are very important. And if I don't put the proper reagents in or the proper amounts in, this step will not happen the proper way. And the whole purpose of PCR is to, number 1, replicate it, but number 2, to actually maybe start breaking it up into fragments as well. That's something that I want to see. Finally, we'll take that DNA sample from the PCR and we'll run it straight on an electrophoresis gel or a fancy piece of equipment if I was working in a laboratory that had one. A lot of this stuff is now automated and I don't really have to worry about electrophoresis sometimes. So here we see an example of a crime scene banding pattern, and then here I see suspect number 1. And I know suspect number 1 is not going to perfectly match, so that suspect was not the one that was possibly at the crime scene when this took place maybe. But if I take a look at suspect number 2, I see almost basically a perfect match all the way across the banding patterns. So that's a very good sign that suspect number 2's DNA was at the crime scene. Did they do the crime? Maybe not, but they were at least present. So why was there blood at the crime scene? Why was there body fluid at the crime scene? Why was there hair at the crime scene? And granted, they might not have anything to do with the murder or the rape or the crime that was committed, but it at least establishes a presence that's actually there. All right? Some things that I want you to take away from the background information. I'll let you read a lot of this on your own, but there are a couple of pieces that I would like to point out and just kind of highlight just for a second or two. And the very first one is this DNA fingerprint. We've talked about DNA fingerprint over and over and over, right? Quite a bit, actually. And you've seen a DNA fingerprint in all of these other labs that we've done. So there's no difference here, right? That's not a big issue. I understand what a fingerprint region is. I know that different fingerprints come from different people based on the order of base pairs that they have. So DNA fingerprint up here at the very top. Something else that I would like to point out is that this technology really began full throttle in the 1980s. So up until that point, it was just mainly blood work, and it was just a few other tests that we did in a forensics laboratory. But DNA was not part of the established kind of routine laboratory analysis that we see it today. And if you look at the dates, 1987 was really the first time that DNA was used in a conviction. And that happened in Orlando, Florida. So 1987, the late 1980s, and now it is so commonplace that this is probably one of the first things that gets done, and it's very easy to do and it's very automated now. Up until that point, this process was very complicated. But keep in mind, the structure of the DNA molecule was actually not discovered until Watson and Crick kind of came along in the picture, and that was in the 1950s, right? So a couple of years later, we finally get it into the laboratory, and then a couple of more years later, we get people that are trained on it really well. A couple of more years after that, we get a better process, and that was this agarose and gel electrophoresis run. And now, up until that point, we have used it over and over and over in multiple cases. Something else that you might not know about is that the Federal Bureau of Investigation, the FBI, actually has this computer database. And right now, it probably has over 13 million different DNA fingerprints inside of the system. And they established this computer system called the Combined DNA Index System. And this is what we call CODIS. So CODIS, if I was working in a forensics laboratory, I would have a login and I would have a password. So I would log into the system, I would key in my password, and then I would upload maybe new DNA that I'm getting from suspects or people that have been arrested or people who are not yet in the CODIS system database. So I would basically submit that information to CODIS, and then CODIS could actually use that DNA fingerprint or fragmentation pattern, and it could cross-reference the millions of DNA samples that it has in the database system at this point. And it can do this from, 10, 15 years ago. So if this person had committed a crime and they had a crime sample that they have a DNA fingerprint of, and this happened 10, 15 years ago, CODIS would be able to use the DNA fingerprint of the person today, cross-reference it, and say, this person's DNA was found on this crime scene dated back in 1991. I mean, those are the kinds of things that we can do now. And you can maybe call it Big Brother and government intervening, but at least that we can do now that will help us process those criminals a little bit better, identify the suspects who were there, and not only in today's crime, but years and years prior. So CODIS is one of those hot topic words. A lot of people do not like the idea of CODIS. They feel that it's the government keeping a track of you, a national database that these people can basically access at any point in time, but your DNA has to be entered into that system. So as long as you're good, and as long as you've not caused any problems, then your DNA is probably not inside of this system. Now, the thing here is that a lot of people, when people like Ancestry came about, or the other DNA companies that you might have read about, and maybe you did one of those tests. You're swabbing yourself, you're providing your DNA to these companies. These companies are basically analyzing your DNA and providing you ancestry traits. A lot of people were very uncertain with these companies because they thought that this was a very easy way for the government to basically tap in to Ancestry.com or these other DNA companies and purchase or require them to turn over their DNA electrophoresis and their fingerprinting techniques, so that way they can start involving this into the CODIS database. So, in other words, if you're at home swabbing your cheek and you just want to know who your ancestors are, maybe eventually that company would then turn around and give the federal government your DNA sample and your fingerprint pattern, and you've not committed any crime at all. But now you are in the CODIS database. So if you do a crime later on down the road, your DNA is already into the database, and they know who you are, and they know your name, and they know your address, at least when you've done your DNA sample. I don't think that they're doing that. These are private companies. They're making a profit, but who knows what goes on behind closed doors. So I'm not going to take a stance on it. I'll just let you make up your mind on if you trust them or not. Okay, so that is CODIS, and that could be something new, and maybe you didn't even know that something like that existed. So again, we'll talk more about that into the lecture videos when we get to that point. Another thing that I want to mention is this prior gel electrophoresis and agarose method. And this was called restriction fragment length polymorphism, Rflp. And Rflp was a way that they could process DNA just like kind of we're doing today, but the problem is that it took forever. And it took a lot of steps and it took a lot of work, and these things could take weeks in order to process. But now we exchange that for a different type of method using different types of reagents, and we can process DNA now within probably 48 hours. A very well organized lab could at least do it within 48 hours. The older method used to be called the Southern Blot. That's what used Rflp. So Southern Blot, if you are reading that or if you see that in some of the literature, this is an older way that we used to process DNA in a laboratory before this gel electrophoresis and PCR came along. So Southern Blot finally got replaced with PCR. It required less sample, it required less time. People loved it, it was great. And once again, we'll talk about PCR in the lecture module when you get to that point. So all of this information down in here, we're going to cover that in more detail in the lecture component of the course. A couple of things that PCR has to have, maybe that I can talk about at least now, is this Taq DNA polymerase. Taq DNA polymerase is basically the initiation step, right? So if you read this first sentence, it says in the first step of a PCR, the template complementary DNA strands are separated, and they are separated at 94 degrees, and the Taq DNA polymerase remains stable. This is actually extracted from the hot springs, basically in the United States, anywhere that has hot spring. And this is an enzyme that can go in and start doing a specific type of job for us. Then it says in the second step, known as annealing, the sample's cooled, usually between 40 to 65. A primer is able to go on, and then you start to replicate the DNA sample. And that's what continues to go on in the third step, is this extension, and the Taq polymerase adds the nucleotides. And those nucleotides are basically the base pairs. So it sits onto the DNA strand, and it starts taking the A, T's, G's, and C's, and it starts assembling them in the order that they need to be assembled in. So that is the extension. So 3 steps for PCR, and that's just a very quick, brief overview by all means. And once again, we'll talk more and more about the intricate details in the lecture module of the class. Okay, so it gives you a diagram of what's happening here in PCR. The good news about it, though, is that we're going to assume the PCR step has already been done for us. So once again, this is just basically pour your gel like you've done before, put your wells in, and then once your gel solidifies, you're going to put your sample in, and then you'll, or samples, and then you'll make sure that they're in the proper order. Make sure that the wells are faced closer to the negative end and the feet of the gel is closest to the positive end. You'll turn your voltage on, and you'll allow these things to run and process. Afterward, this will require staining because they are kind of DNA samples, but they're just not from a person. So you will see your particular bands here like you saw in your previous electrophoresis experiment. They will not be colored blotches. That was only the first one that we did to get you used to it. So it does require a little bit of staining. You'll have to have time to destain it so that way you can see it a little bit better. But that same process is basically identical to the last experiment that you did. The only difference is that we're comparing crime scenes to suspects, and we want identical matches at this point. We do not need to look at mothers and fathers and do parental side of the house like we've done before in the other 2 experiments. All right. Again, 60 degrees is pretty important. That way you don't destroy the molds that are on the casting trays. That rubber will start to disintegrate and get crusty over a course of time, and then before you know it, these things are going to leak, and people are not going to be able to pour their agarose in because that casting gel is not, or tray is not going to hold their gel. Recipes, just like before, are down here in the bottom. We are typically using the 7x7, so it tells you how much buffer and how much water and how much agarose to weigh out in order to make the 0.8% agarose gel that you need to use in the laboratory. Next, they're giving you a diagram of the buffer. Again, this is 1 times diluted buffer, so they will go through and give you directions on how to make this buffer down below. But again, we have different trays, not Edvotech trays, so we'll have enough buffer at least to fill all of your trays up, and then that way we can turn the juice on and your DNA samples can start to run through the gel. Tube A, Tube B, C, D, and E. How are these in order? Well, Tube A is your standard DNA marker. I'll show you what that is in just a second. Tube B is the crime scene PCR reaction. So the PCR piece has already been performed for us, and we are just analyzing the DNA at this point. Tube C, D, and E are your suspects 1, 2, and 3. And again, those are PCR reactions. So the PCR step has already been performed by someone else. This is kind of the end result of that process, and we are injecting it onto a gel, and we're just letting it band out. That's all that we're doing. So keep in mind that PCR has been done prior. So this would have been extra work that you would have had to put in if it wasn't for that. And that would have taken at least a day to 2 days for us to do. And you're eventually going to do it. You're just not going to do it yet. So this is another training exercise basically to get us ready for that particular step when we extract our own DNA from our mouth. All right. If you keep flipping through, it's going to tell you how to stain and destain your gels. Just like before, there's really no difference. They give you a couple of options. So we're going to have this stain that we're going to mix and dilute down a little bit. We'll slide our gels down into the staining trays. We'll let it sit for a few minutes. It says 5 minutes here. And then we'll pour that out and we'll flush it with some water. And we'll keep flushing it with water if you need to in order to see those bands maybe a little bit better. So it gives you a couple of options, but there you go. The purpose of the stain is to stain the fragments pieces, so that way you do not need anything special to look at the fragment pieces. So we can visualize these with just normal light in the room. Not a big deal, not a problem at all. You should be able to see those bands, and you should be able to take a picture of those bands as well. Here's just another way that we can stain. This uses the paper instead of the liquid. Either way is okay. It really doesn't matter. Honestly, it's just whatever we have that we bring out into the lab that we'd like for you to use at that point. And then the last page are going to be a couple of questions. What is polymorphic DNA? How is it used for ID purposes? What is CODIS and how is it used to solve crimes? And what is an STR and a VNTR? This I did not talk about, but it's given in the background information. So if I want you to answer these questions, these will be on your post-lab questions document that you will receive from me that is uploaded into Blackboard. So if these questions are not in that document, do not answer those. But if you do see questions like this in the lab document, then my advice is to go back to the background information that we talked a little bit about, and you'll be able to see all of those answers basically right there in that narrative, okay? So I think that this will be fairly easy for you. You've already had 2 trainings up to this point. This is going to be your 3rd training on electrophoresis, and it's basically the same stuff. Pour a gel, put your samples in the wells, put it into the thing, pour your buffer in, and then crank the voltage on. And you'll see the banding patterns that will happen because of that. So I think that you'll be okay as far as the lab technique goes. This is just another different perspective about electrophoresis, this time comparing crime scene samples to suspect samples. That's all that we're doing. So I hope you have a good time with the lab. Again, I think it'll be an easy A for you. And then after this one, I promise, we'll start to extract real DNA, and you'll see all of the process that goes on from point A to point quadruple A. Have fun.